I presume you were clapping Antoine, but if it was my entrance, then I'm very pleased. So let's see if I can do this. How's that look? So for those of you who know nothing about hemoadsorption, this might be quite informative. For those of you who know a lot about it, just fantasize about something else for 15 minutes while I go through the basics of hemoadsorption. So what I'm going to talk about is, first of all, my declarations of interest, and I've had payment to my institution for research from Exthera Medical, who are a company that make the Seraph column. So I'm going to talk about the ADQI conference and the distillation of that meeting on absorption-based therapies. So some of you may not be aware of ADQI or what they do, so whistle-stop tour of that. ADQI, Acute Disease Quality Initiative, was originally the Acute Dialysis Quality Initiative, but has changed over the years. It's a non-profit, member-run organisation where we write dispassionate distillations of the literature, not always completely evidence-based, sometimes expert-based because the evidence isn't there, but we focus on areas of critical care nephrology, really. And some of the things you'll be aware of, like this was the second ADQI conference, and from which the rifle criteria came from, and the subsequent definitions of AKI from KDIGO were triggered by the ADQI congresses, of which we've covered an array of acute kidney injury topics. And you can see this particular paper has been cited over 8,000 times, and I think is still the most cited paper from critical care. So this was the 30th ADQI conference, and it was in Vicenza last June, and it was focused on absorption-based extracorporeal therapies. And the idea was that we would develop consensus and recommendations for taking these treatments forward. And as you can see, these are the countries involved, so there was a fairly good spread across the world. I could quiz your flag knowledge, but I won't, right? Just say we involved countries from around the world. Now, at this juncture, it's probably worth considering why we're here, right? Because some of you may be naysayers in the audience saying, well, yeah, but hemoadsorption doesn't work. Why are you doing it? And, you know, there's no positive trials. Some of you may be advocates and saying, it's fantastic, you know, that's what we should be doing to all our patients. Well, I can warn you that I'm somewhere in the middle, right? And I think that this particular area can be an area of great excitement, and we need to be a bit more positive, right? And we can design trials in the future that will help. The cardiologists, for example, won't be quite so naysayer-like about their technologies. So what were the specific objectives for this conference? Well, here they are, but they can be distilled to four main questions. There was the rationale and the physicochemical principles behind hemoabsorption. Because some of you may think, well, it's just like dialysis, isn't it? Well, it isn't, right? And then the techniques and modalities of absorption-based therapies, you can see I chaired that bit. Then the potential indications, prescription and monitoring, and then the endpoints that we might use for studies. So I've already used the whistle-stop tool once, so I won't say it again. So here's the group one, rationale and physicochemical principles. So basically, hemoabsorption is an extracorporeal therapy, and its aim is to remove solute cells and pathogens. So you may say, well, that's similar to other renal replacement therapies. Yes, but it's aimed, due to physical chemical features, you know, of things that we're trying to remove that are not amenable to standard dialysis or filtration convective clearances. So what does that mean in practice? Well, it means that hemoabsorption represents the dynamic binding of these absorbents in the blood to a solid surface. And that's achieved by you pass your blood through the sorbent, through the cartridge, and in contrast, the removal of substances by absorption means the blood has to touch or come in contact with the biocompatible absorptive material. It forms a bond in some cases and is then removed. So this diagram kind of cartoon will explain that better than I do. And so, interestingly, the video didn't work. Well, that's a bit of a shame. Oh, yes, it did. So these are small molecules that we're not interested in, right? So we can see they will pass through the column and not absorb. Whereas we have target molecules, of a different size will undergo absorption to the medium that we're using, and then larger molecules that we're not particularly keen on removing, like red blood cells, are not removed. So it's dissimilar to dialysis, where you're using mass transfer across a membrane, or filtration, where you're using convective treatment. And you can imagine that you can change the components of your column to remove certain things that you may be interested in. So polymyxin, for example, removes EAA. So this technique absorbs middle to large molecules and protein-bound substances. So this is the kind of picture, these beads that are within this medium that the blood passes through. Now, you can use hemoadsorption in the setting of conventional dialysis, and you can use beads with very large surface area to volume ratio to target very specific molecules, and the opportunity to remove greater volumes of those solutes than you would otherwise. So how do these columns work? How do they do that? Well, you can either have direct chemabsorption. So you actually get chemical bonds between the solute and the sorbate, right? Solute and the sorbate. Or you can have physical absorption. So that's water-based interactions, hydrophobic interactions between the solute and the sorbent, or the more mysterious ionic interactions. You may remember these from your high school chemistry, Van der Waals forces and things like that. So these three physical-chemical principles dictate how these columns work. And one or more of those mechanisms may be involved when you're using it in a patient. And it depends on the absorption you're using, the device configuration, and things like that. So absorption, hemoadsorption, is based on certain basic principles. Right? The solute reaches the binding through mass transfer, so it goes along a concentration gradient, and that's influenced by flow rate, blood flow rate through the column, as well as the solution characteristics. And then active binding is within the pores themselves, that kind of mesh I showed, and that's a crucial determinant of access to the sorbent. So if your thing is too big, it won't pass through the pores to get to the sorbent. So, technical design that the companies like Jafron put into these development is quite impressive. And then when solute reaches the potential binding site, the absorption is influenced by the factors related to the solute themselves. Now, the dialysis geeks in the audience, if there are any, may recognise these isotherm equations. This is how we quantify binding of particular substrates to a column, particularly in dialysis, but isotherm equations are used throughout industry, not only in renal replacement, but in other physical processes. And so we can describe the capacity of the filter by its isotherm, and also by using the removal rate of something over time, which we're far more familiar with. And the selectivity of the absorption is influenced by the membrane itself, the device configuration, and the conditions it's used under. And selectivity depends on the device and the patient features. So it's important to realise that they're not all the same, and they all have very different characteristics. They may have a generic ability, but some are specifically designed to remove certain components. And so, as I put there, removal of endotoxin with polymyxin B is something that you may be aware of, or the removal of broad-spectrum cytokines, for example, is seen in other absorptive columns, such as Cytosorb, as well as some of the other Jafron columns. Now, if we describe the techniques and the modalities involved, the modality is the method by which the blood or the plasma is exposed and removed. So endotoxin removal will be a modality. Removal of bacteria or viruses from the Seraph filter, the heparan bonding filter, will be a different form of modality. And the technique is how it's removed via an extracorporeal circuit. So that's slightly different to what you may be used to. So these are examples. Now, it's important to realise that a lot of the things listed on here, this is from the publication that will come out in a few months' time, are not CE marked, so they're not available for human use at present, but it shows the degree that this field is changing and how it's shifting. So how can we use these things in practice? So the simplest circuit is a straightforward hemoadsorption circuit. So some of you may have seen the Smart machine made in Italy, which is a simple hemoadsorption single pump unit. I mean, that's what I tend to use. It's easy to use, and you know what you're getting is just the effect of the filter, rather than when it's in conjunction with other things like CVVH, CVVH-DF, or indeed intermittent hemodialysis. So these things can be fitted into these circuits, either pre or post filter. It's important to bear in mind before you just buy one, pick it out of the packet and plug it in, that you know how to set it up on your machine and how it runs. But most of the machines will have the ability to do that. The alternative is you can put it in a bypass circuit. There's some exciting work coming out looking at patients on bypass, or indeed an ECMO circuit. Historically, hemoadsorption has been around for many, many years and was originally used for exogenous or endogenous intoxications. So reducing endotoxins, for example, reducing unwanted therapeutic effects. So there's good evidence now for clearance of things like apixaban by absorptive columns, or microbiological effects, removal of viruses. The Seraph filter has some impressive data on the removal of COVID and improvement in patients treated with that. The American military are one of the few that use the Seraph column routinely, and that's why you may remember the erstwhile president got removed to one of the military hospitals when he had COVID. He'd obviously run out of bleach to clear his COVID, so he thought he would use something more scientific. Problem is, with a lot of the studies, they're of low quality, many are retrospective, and of single-center design. And so, as a consequence, I think that's reflected rather unfairly on the field in general. Now, how do we indicate the indications, prescriptions, and monitoring? This is another picture from the paper. It's clearly an excellent figure, and part of the reason it's an excellent figure is because Antoine designed it, but it shows the different indications, such as poisoning, sepsis, or a cardiac procedure, and how they may be integrated within. Another example can be that of rhabdomyolysis. There's quite good evidence now, and there are papers that are going to come out soon, consensus statements on the early removal of myoglobin and improvement in outcome in these patients. And remember, something like myoglobin isn't filtered, it's poorly dialyzed, but an absorptive column will wipe out your myoglobin concentrations quickly. What about starting, and when do we start treatment? Well, there isn't enough evidence for us yet to guide the start of hemoadsorption. As I said, there are some studies on cardiac bypass which will come out. That's a fairly well circumscribed treatment. Right? Patient goes on a bypass, that's when we start it. But it should be based on the pathophysiology of the underlying condition, where criteria suggest that the clinical course may be modified for therapy. But it shouldn't be used as a last resort Lazarus treatment. Right? We've got nothing else, so let's try the filter. Because if we do that, we just won't progress. I think that's an important take-home message. So, endpoints. What should we base our studies on? What should the endpoints be? Well, the phase of development of the illness, the clinical indications, the specific of the treatment used, and the intended patient population. And this is all related on disease and context-specific factors, like most of the studies we do. If we design a poor study, we're going to get a poor result that's not translatable. So hopefully, in the next few years, we'll get better studies with outcomes that will inform us in terms of our management. And so, you may remove a specific disease, a specific biomarker, or physiological toxicity may be one. So we may see a biomediator that we want to remove from a patient, and that's reflected in a biomarker concentration which we can measure, and that might be the endpoint. So to conclude, there's a strong rationale and much evidence about modern hemoadsorption, how it works and what it removes. It's clinically acceptable, it's biocompatible and safe, technically it's feasible, and there is experimental evidence of target removal, particularly when you're looking at drugs. Pilot studies demonstrate beneficial effects on biomarkers and physiology. And larger studies of endotoxin-based hemoadsorption have identified potential phenotypes, right? So not one size fits all. And in a variety of intoxications, removal had been confirmed. And I think that's certainly an area of growth. But despite these achievements, it's still a relatively novel intervention for us with a limited randomised controlled database and a very large research agenda. And I think that that's the bit that should get us all excited. And with that, I'd like to thank you for your attention.